behalf of the President of Children's Education Society and Chairman of Oxford Educational Institutions, Management, Principal, Staff and Students of the Oxford College of Engineering, I, Dr. K. Valarmati, Associate Professor, Department of Biotechnology, deem it a great pleasure and privilege in welcoming you all to the 5 days International Virtual Symposium on An Insight into Biotechnological Application. This virtual symposium brings together experts and students from science and engineering and research community and explores the technological advancements in biotechnology engineering. The Oxford College of Engineering, TOCE, is one of the most prestigious institutions run by the Children's Education Society. TOCE aims at carving competent students with extraordinary skill to face challenges of the continuous changing world. TOCE provides quality training in professional education and values in engineering, management, and computer application streams. The college has 11 research centers approved by VTU to carry out PhD and MSc engineering programs. The institution is well known for its consultancy and research and development activities. Various departments have financial assistance from reputed funding agencies, namely VGST, KSCST, VTU, DRDO NRB, DAE, BRNS, AICTE, ICMR, National Innovation Foundation, DBT BIRAC, such as and funding agencies to carry the need of young researchers and students in engineering and management.
On behalf of the Department of Biotechnology, on behalf of the staff, students, and management of the Department of Biotechnology, the Oxford College of Engineering, we welcome Dr. Joy Harris Huskari for this International Virtual Symposium. Dr. Joy finished his PhD in bioinformatics on research entitled Proteomic Analysis and In Silico Drug Designing Studies on Hepatoprotectivity of Herbal Constituents isolated from Flavaria trinervia C. Moher, carried out at Department of Biotechnology and Bioinformatics, Colombo University. Sir is currently working as Assistant Professor at Department of Bioinformatics, Akkamahadevi Women's University, Vijayapura, Karnataka, India since 25/5/2018. He also worked as a visiting scientist in Laboratory of Molecular Pharmacology and Biophysics of the Institute of Experimental Pharmacology and Toxicology, Center of Experimental Medicine, Slovak Academy of Sciences, Bratislava from 21/6/2019 to 21/7/2019. His achievements: he was awarded first rank in MSc Bioinformatics from Department of PG Studies and Research in Biotechnology and Bioinformatics, Colombo University, India. And then he was been awarded Young Gun Award in a CSIR sponsored national conference at Tumkur University, Tumkur, India. He has received many best poster presentation awards in various national and international conferences. He has a total of 56 research papers published, and he has presented almost like 39 research papers in international and national seminars. He has published 2 books, and then he has published 4 book chapters. So I would like to take this opportunity to extend a very warm welcome to Dr. Joy for this international 5 days virtual international symposium. And also I would like to like to welcome our principal Dr. Aravind A.S., our director Dr. Amarnath K., and all the participants all over India for this 5 days international virtual symposium. Also, I would like to welcome Mr. Mukund, alumni of our department for this webinar. And also, I would like to take this opportunity to welcome our HOD, Dr. B.K. Manjunath, for this webinar, and then all the other faculties and students from all over India. Welcome all. So, now we will be starting with this session. Joy sir, can you please share your screen?
Yes, ma'am. Thank you. visible? Host, is the slide visible?
Not yet, sir. Can you? It's loading. Sir, can you stop sharing and then try once again sharing? It has not come. Sir?
Yes. There's some problem with the sharing.
Yeah.
It's taking time. Is the slide visible?
Yeah, sir. It's visible. It's visible, sir.
Okay. Can I start?
Yes, sir. You can start.
Good morning, everyone. I'm Dr. Joy today. My talk will be on proteomics and biomarker discovery. Here we are specifically focusing on protein biomarkers. So biomarkers, what are biomarkers and what is proteomics? What are the tools and techniques that we use in proteomics to identify potential biomarkers for various diseases usually
Sir,
Proteome
can you speak little louder? It's not audible.
proteomics. Proteomics is a large-scale study of proteomes. Now, what are these proteomes? Proteome is a set of proteins produced in any organism or a system or any biological context. It might be tissue or cells or a group of cells or organ or an organism. Proteome normally is not constant. As you see, if you see the protein content in one part of the body, you check the protein content in the another part of the body, that is different. If you check the protein content in your skin and at one particular point of time, and after some time you again check the protein content in the same tissue, there will be difference in the total protein content. The reason is there is fluctuation, there is upregulation and downregulation of protein content in the same tissue. So proteome as a whole in any given system is not constant. There will be fluctuation in the protein content in that system. So it's very important when we are studying the protein content, the total, all the proteins present in a given tissue or cell. It's important for us to study them at a one particular point and then understand what is their expression level in that particular tissue. And it can apply to the same tissue or it can apply to the normal versus diseased tissue. So to some degree, the proteome reflects the underlying transcriptome, or I would say all the mRNA content in the given tissue. Some extent or some degree it's going to reflect this has a meaning. Why is it mentioned that to some degree the proteome reflects the transcriptome is because not all the transcripts may give rise to the protein, number one, and the same transcript can be responsible for producing different types of proteins with different functions through post-transcription translational modification. Well, proteomics can provide significant biological information for many biological problems. Mainly, here we are using proteomics in understanding the molecular pathology that takes place in the system due to any disease and try to identify a candidate biomarker for that particular disease. So there are several technologies in proteomics. So proteomics is, I would say, it's a cutting edge technology now where we use it for studying the expression levels of all the proteins in a given system. So most commonly applied is mass spectrometry based technique. Especially we use tandem mass spectrometry. I will be talking about that. And also certain gel-based techniques where we use for resolving all the proteins expressed in a given tissue. It might be dye-etch or differential gel electrophoresis, or it might be two-dimensional gel. Now, these high throughput technology, why do we call them as high throughput? Is because these are the technologies that generate a lot of information. So when we perform an STS page, we may resolve 100 to 200 proteins in a gel, whereas two-dimensional gel process has the capacity to resolve 300 to 11,000 proteins, but practically speaking, it can be at the most 600 proteins can be resolved in a technologies where we use mass spectrometry, light track technology. We can generate lot of information about all the proteins that are expressed. Now I am repeatedly using the word expression of protein. Now when I say expression of protein, that means proteins have a very significant role to play in our body. So once we have generated large amount of data, now it's important for us to store that and retrieve that data from a particular stored place, and we call it as a database. So databases are critical for recording and carefully retrieving and storing. So proteomics in biology, when you see proteomics can be used in expression profiling, where we study all the proteins that are expressed in a cell or a system. Or proteomics can also be used for post-translational modifications, where proteins undergo in order to become functionalized. Proteomics can also be used to study protein-protein interaction in order to generate an interactome map, or can be applied for studying protein structures, what we call it as structural proteomics. Proteomics can also be used in protein quantification, where we can measure the expression levels, whether some proteins are highly expressed and some proteins are lowly expressed, what we call them as upregulated and what we call them as downregulated. Proteomics can be used for studying the protein functions also, what we call it as functional proteomics. And it can be a part of proteome mapping or mining, where we try to study and collect information regarding various proteins of the system. What are biomarkers? A naturally occurring molecule, it might be a protein, gene, or a metabolite, by which a particular pathological or physiological process of a disease or a condition of human body can be identified. So these are the indicators. I would be specifying. There is another definition. It says that in medicine, a biomarker is a measurable indicator of severity or presence of some state. More generally, a biomarker is anything that can be used as an indicator for a particular disease. For example, if a person is diabetic mellitus, suffering with diabetes mellitus, and for a diabetic patient, glucose level is a indicator, right? So we measure the glucose level. If the glucose level is too high, we say that the patient is diabetic. So glucose is considered to be a biomarker. So it can be a metabolite biomarker here because it's glucose. Similarly, we have various proteins also as biomarkers. If a person is suffering with hepatotoxicity or liver injury or fatty liver, so we tend to go for liver function test. Then we study SGPT, SGO. That they might be a measurable damage in the liver. So most commonly, many people consume paracetamol. That is, you might be familiar with the various brand names for that, Crocin or Dolo 650. And we all are familiar that the paracetamol that is a liver toxin. So we, for the fever, we take that particular medicine. And most commonly, people consuming that on excess amount will suffer with fatty liver. And we use liver function test to find out that has happened with hepatotoxicity. And there we use protein markers. How did we come to know that these are the proteins that has to be studied to find out the disease, the type of disease the patient is suffering with? Now, we have concluded that the glucose level, if it gets increased in the system, then we say the person might be suffering with diabetes mellitus. Now, the point is, we have to find out a biomarker, a significant biomarker for various. There are many diseases today for which the biomarkers is still not been identified. And there's a need to find out potential biomarkers that can reflect the severity of it. So let us focus on protein biomarkers because today's topic is on proteomics in biomarker discovery, and obviously we'll be focusing on protein biomarkers. So why proteins? Why proteins are so important? So if you see, proteins are so important that your entire physiology, your function is totally dependent on these. You should know that proteins in our body, there are millions of proteins that are functioning in our body. So if I'm talking and you're listening, your auditory receptors in your ear is receiving the signals, and you are noting your head is because of the proteins that are functioning with which your body muscles are moving. If you want to raise your hand, you should know that there are millions of proteins. If you are hungry, the hungry response that is going to the brain is because of the thousands and millions of proteins that are functioning. If you touch a surface which is hot and you just remove your hand, the impulse from the finger, it goes to the brain, and from there the impulse goes back to the hand, and if you take out your hand, it indicates that there are thousands of proteins that are functioning in your neurons and in your muscle cells with which you are doing certain activities. So proteins are very active in your system. You should understand, if you are lazy, your laziness is also because of those millions of proteins that are in your body that are actively functioning and making you lazy. So proteins are there, proteins are functioning, and because the proteins are functioning, we are functioning as a complete organism. So on the other way, I would say that if there is any fluctuation in any protein expression, we will suffer with it. Here you can see the proteins have just bought some animation. So proteins are playing very important role not only in metabolic pathways but also in signaling pathways. Why proteomics is so important nowadays? You can see there are various publications coming up. Human proteome, after human genome project was completed, the proteome project has been up and now it's in the process now. I think they have already covered a lot of proteins now, their functionality, their uses, and their applications in various diseases, cancer. Now, why so much of investment is being made on this understanding the entire human proteome? There are many other literature also published on human proteome. Now, the reason is proteomics has given us a very important clue to understand diseases in a much more better way. Now we can understand the molecular mechanism of any disease through understanding its proteome, its expression level, what proteins are upregulated, what proteins are downregulated, we can understand. Okay. Now, for in proteome project, the entire human system was covered. But here, in the context of diseases, we will be specifying only a specific disease and we will be using it in understanding the protein expression in that disease condition to check which proteins are upregulated and which proteins are downregulated so that we can find out which are the key proteins that are differentially expressed. So when you see proteome is different from transcriptome. The reason is when you look at the complexity from genome to the protein level, from genome we get a specified if there are in a genome, if we say that after human genome project we have realized that there are 25,000 genes so far identified. So it's not that 25,000 genes will give rise to only 25,000 transcripts, but 25,000 genes will give rise to more than 1 lakh transcripts. And these 1 lakh transcripts will give rise to more than 1 lakh protein. That is set of all the proteins is a protein. And these proteins will further undergo post-transcript translational modification through which they will get different functionality. Now you see this is the same protein which was translated, but during post-translational modification, the same protein can be hydroxylated, it can be tagged with ubiquitin tag, lipidation or zumylation, it might be phosphorylated, acylated, methylated, glycosylated. Through this, the same protein can get different functions. So we should understand the complexity of the proteins that are there in the body because what we talk about the number of genes that are present in the genome versus the different types of proteins that we see in a system is far different. Okay. So when you see an interactome, different types of proteins that are interacting in a given system, you can see these dots that you see here. These are the proteins that are interacting. Okay. I would like to show you one more image where you can see our entire interactome map of human looks like a constellation of stars. So that is the complexity of the proteins that are actually functioning in your system. So in order to decipher or in order to understand each and every individual proteins that are expressed in the given system, it is important for us to follow certain technologies. So we have various technologies now. Currently, we can understand them. These are all advanced technologies. So today, I'm going to discuss a couple of them, covering its importance in biomarkers. Am I audible?
Yes, sir.
Host, am I audible?
Yes, sir. Hello, sir.
Am I audible? Am I
Yes, sir.
audible?
Yes, sir. I think some slight break, but otherwise it is audible.
Okay. Am I too fast?
No, 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 sir.
Okay. Fine. Okay. The steps involved in biomarker discovery. Here again, I'll be specifying only on protein biomarker. through proteomics approach. Firstly, whenever we going to find out, if we are intending to find out any protein biomarker for a particular disease, let's say in context of cervical cancer, if you are intending to find out a biomarker for cervical cancer, if you want to use the urine of the cervical cancer patient and find out a particular protein that is abnormally found in only in the cervical cancer patient and not in the normal patient, normal person. So in that context, you are going to take the sample, the urine sample of the cervical cancer patient versus the normal. Now, you need to find out what are the different types of proteins that are found in the urine sample of cervical cancer patient versus the normal. So that you can differentiate and find out what are the differentially expressed proteins that are found in the urine sample of the cervical cancer patient. Now, in order to find out that, first of all, is to do the protein separation step. Now, you know that the urine is composed of various metabolites, chemicals. Along with that, proteins are also part of it. Now, separating the proteins from that sample is very essential. It cannot be specifically unique. You can also go for, if you are looking for a saliva-based biomarker, if you are looking for blood-based biomarker, or if you are looking for any tissue-based biomarker. It depends on what is the sample that you are going to work on. So any sample, you can take a tissue, you can take the blood, or you can take saliva, or you can take the viscous fluid, or you can take urine. Any sample that you're looking for, first thing is separate protein. So you know that samples are composed of lipids and other metabolites, nucleic acids, proteins. And it's essential for us to do proteomics. It's important for us to separate proteins in the process. The first thing is protein separation, followed by mass spectrometry, where we will be studying, or what you say, sequencing proteins, understanding them and annotating them. After that, we will be performing bioinformatics, where we understand the functionality of the proteins and in which pathway they are involved and how they are related with the disease that is under study. A general overview, you can see it like, first thing is sample. We take sample and we do fractionation. Then we go for performing a gel-based separation. After we do gel-based separation, and if you're looking for some specific protein that you are interested in, then you can separate, excise the protein, and digest the protein using trypsin. Once you get the fragments, these fragments are then put to mass spectrometry. Here we do mass spectrometry. So through mass spectrometry, you're going to get a mass spectra, which will be reflecting different peaks, and through peaks, you'll be able to identify what is the fragment, which fragment, what is the sequence of amino acid in that fragment. So this is general overview, where I'm talking about SDS page, but today I'll cover about 2D page also. Just we'll go step by step. Steps covering, as I said, first is sample preparation. In the first step, any tissue or sample that you are looking for, find out a potential biomarker for a disease, you have to first consider what sample are you going to work on. The importance of the sample here. It's very important for any patient to give the sample very easily. You cannot go for tissue biopsy every time. If you find out a biomarker in a tissue, you cannot cut the tissue from a patient every time. But the samples that are easily accessible, for example, blood, saliva, urine, working on such samples are most important, because the biomarker can be potentially being used in such samples. Otherwise, taking the tissue, it is like the kind of tissue damage that the patient undergoes. Patient normally avoids giving the biopsy. So sample that we are choosing for proteomic analysis is very important. The whichever sample we take, we need to separate the proteins from that sample. After we separate the protein, next comes studying the proteins that are what you find is they are differentially expressed, and then subjected for mass spectrometry. Sequence those proteins and find out what are those proteins, what is the functionality of the protein, and how is it involved in the disease. And if you find it as a potential biomarker, then you have to use it. Now, coming to proteins or proteome sample preparation, there's a difference. Protein sample is different. Proteome sample is different. When I say protein, I'm talking about a single protein. When I say proteome, I'm talking about a set of proteins. I mean all the proteins in a given sample. So sample preparation, the process goes with extraction, okay, then salting, concentration, and fractionation using strong cation exchanger. The sample can be cells, tissue, organ, or organism, whichever sample you can choose to work on. Once you take the sample, you need to homogenize it. After you homogenize, next comes the process of salting. During homogenization, we can use methanol, and for salting out, we can use ammonium sulfate or ammonium acetate salts, salt solution. When you saturate this, the sample solution, the protein starts getting precipitated. After you add the salt solution to the sample, it has to be stored in cold, minus 20 degrees centigrade overnight, and then we see precipitation of the protein. Then spin down the proteins so that you get the concentrated proteins in the precipitate. You can exclude the supernatant, wash thoroughly the pellet, and then that is the complex mixture of all the proteins in the given sample. Then comes the protein isolation. If you are looking to work on only single protein, you can go for isolation. If you're working on all the proteins, then you can go for cleanup using resin packed micropipette tips. You can clean them up and then go for fractionation. Fractionation, normally we use a strong cation exchanger where we fractionate the, the mixture of proteins, different fractions, and whichever fractions that can be pulled up, we can pull it. The main reason here is to remove the contaminants from the sample that we have. So the cleanup goes where general purpose cleanup, where we just pass them through resin. Whereas fractionation, we pass it through a strong cation exchange. Once we get the fractions, we then put them wherever it is required. Then comes separation. Separation of the proteins uh, can be done by either SDS page or two-dimensional generative process. If the protein content in your sample is measurably low, for example, in tears, that is the vitreous fluid, or it can be in urine, the protein content is measurably low. In that context, you can use SDS page. Or if you're working on blood or tissue, in that context, you can go for two-dimensional fluorescence. So you all are familiar with SDS page, that is sodium fluorescent sulfate polyacrylate gel process, where we use SDS, which is ionic detergent. It goes and makes the protein negatively charged, and as we run the set of proteins in the gel, they get separated based on their molecular weight. So this is a common technique that we all are familiar with. So once we separate the proteins in the SDS page gel, we get the gel profile in a band form. Right? So there we can visualize and compare the normal the gel pattern, I mean the band pattern versus the diseased condition band pattern. And if you are looking at some difference in the band levels if, compared to normal, in disease condition, the band is very thick and increase. There is increase in that protein concentration in that disease condition. Or in some context, it might be very low or very faint band, indicating the concentration is low. Now, here we can find out which protein is highly expressed and lowly expressed in this condition. Now, this is what makes us to find out why do we consider them as a biomarker? Because such proteins which show increased expression in the disease condition, those proteins are, we can consider them as an indicators or biomarkers for that disease. Because during the disease, that protein gets upregulated. That protein expression will be more, and you find a high concentration of that protein in that disease. So SDS page can be appropriate for low protein concentration. But if you are working on in tissues where you see a lot of proteins, where thousands of proteins are found, in that case, it's important for us to perform two-dimensional gel electrophoresis, where the first dimension is isoelectric focusing, and the second dimension will be a common SDS page, where we separate the bands based on their molecular mass or molecular weight. So isoelectric focusing is totally based on the isoelectric point of every protein. So it's a point where it's a pH at which the protein becomes neutral. That's its isoelectric point. So here we do, in the first dimension, we mix the protein sample with the ampholytes. Ampholytes are like uh, different buffer agents which can set the pH gradient in the first dimension strip. We mix them and run the first dimension on a strip-based method. We have IF strips or IPG strips, isoelectric focusing strips, where thin plastic will be coated with the gel, and we load the gel with the uh, protein content along with the electrolytes, and we run that gel for for a long time. Let's say from 4 hours to 16 hours to 36 hours. We run them um, at a high voltage. So once we run them, the proteins get separated based on their isoelectric point. So after they get separated based on their isoelectric point, we take the same gel and then we put it on the second dimension SDS page. So once we place it on the gel and we start running the second dimension, the the bands that was separated in the first dimension will start moving to the second dimension gel, and we get dissolved gel profile. So once the gel is done, we perform staining. Okay. The staining can be either silver staining or Kumasi blue staining. For SDS page, we use silver staining and Kumasi blue staining. Silver staining because the even the smallest expression can be found out through silver staining, where Kumasi blue staining of the faintly expressed proteins will be masked or not visible. So in that context, we use silver staining in case of SDS page. So Cypro also, I have used Cypro. It also gives the color like dark color, dark brown color. Radioisotope labeling nowadays is stopped only if required to perform. In two-dimensional gel electrophoresis also, we use the same type of techniques, but we use something called fluorescence staining, Psi-5 and Psi-3. So using fluorescence staining also, we can find out the differentially expressed. Normally, a two-dimensional gel electrophoresis gel profile looks in this way, where you see spots of proteins. Okay. Some proteins may appear intense, and some may be very small, and some may be minute. Now, the reason this indicates the expression levels of the protein are high. Okay. So these are low, and the smaller ones, what you see here, they are very low. I would say this is one of the best profiles where you can visualize the protein expression. Various proteins are able to see profile. So normally, when we want to compare the proteome between two systems, we check the expression level, the location, the same location. You have to check it in the. If this is a normal state, if you say this is a normal state and this is a disease state, you have to run two gens and then compare. The same spot, you can see this here is a protein. The same spot you can see one more protein. This indicates that this protein is upregulated in the disease condition.
Excuse me, sir.
Similarly,
Sir, excuse me, sir.
yes,
Your
yes,
voice is breaking, sir.
okay, yeah. Now,
Yeah, it's fine now.
yeah. So, in some context, we can see that here is a protein. This protein is not found in this condition. That means this protein was absent. This protein was absent in this condition. This is absent. Similarly, here you can see there's no protein, but here you can see there's a protein. Here there is no protein, indicating that the protein is absent in the normal condition, but it's found in the disease condition. So such proteins, what you say that they are abnormally found in the disease condition, and they have to bioinformatics. Once we find out that these are abnormally expressed, then we will be adding these proteins. We will cut this protein from the gel and then, sorry, okay, specifically, if you are looking for some protein, you will be cutting this protein from the gel and then will be uh, electrolyting this particular protein and then cryptic digest and do the sequencing using mass spectrometry, find out the name of the protein, find out its functionality, its involvement in the disease, and then we can conclude if it is a potentially involved in the disease, then we can conclude that this is a potential biomarker of the disease. So it can be if you want to study, you can compare it between two different conditions, two different cells, or disease versus normal. You can do proteomics work in any condition. You can take a hair follicle, a white hair versus black hair, the gray hair versus white hair. You can check the hair follicle. You can study the profile. Okay. You can also study the tear, the tear of a laughing tear versus a crying tear. You can take both, and then you can do a comparative analysis. And you can do proteomics. So here, you see, here is an example that mapsin protein is found in a diseased condition. It's not found in normal condition. That becomes mapsin becomes a potential marker for a particular disease. If it is expressed, it is expressed only in that disease condition. So you can see. Normally, we do comparative studies in biomarker identification. Tight differential gel electrophoresis where we don't use two gels, we use a single gel, but we pool the samples. Like for one sample we add sci-fi and the other sample we add sci-fi fluorescent dye, and we pool them. So a green coloration indicates the set of proteins from normal sample, and the red color indicates the set of proteins from the disease sample. If there is mixing and giving yellow color, that means both the proteins, in both conditions, the interest equally. If the red is intensified, it indicates only in the diseased condition that proteins are found. So dye can be performed on a single gel using fluorescent dyes. I told you, after we cut the gel, once we find out that there is some protein, let's say if you are looking for this protein, and you found out that these are the proteins, some 10, 20 proteins you located, they are abnormal. So you just cut them, cut the gel, extract the protein, go for digestion. You need to break the proteins because mass spectrometry has a problem that it cannot resolve high molecular weight protein. So for that, we need to cut the proteins into fragments, and we can then resolve the fragments because mass spectrometry, the limitation of the mass spectrometry is 4. Now, here we digest the proteins and using trypsin. Trypsin is a proteolytic enzyme. So the serine protease cleaves the protein at carboxylic end of lysine and arginine. But if these amino acids are being followed by proline, then it doesn't digest. After we digest, we subject it to mass spectrometry. Now, mass spectrometry, here you can use LCMS, or you can use Maldi MS, or you can directly use mass spectrometry. Depends, right? If you are looking for LCMS, we need to load these peptides that we get from trypsin digestion. We get peptides, and these peptides will be loaded onto the mass spectrometer. So liquid chromatography is a technique for separation of mixtures. Here the mixture is a mixture of peptides. Right? So every time, every gel spot, what you have cut, you will be running a different mass spectrometry for each one of them. So if you are studying hundreds, so you will need to run 100 of spots separately, okay? Separate mass spectrometry. So highly sensitive, you know that LCMS is highly sensitive mass spectrometry, where it can provide the information for identification of peptides. LCMS is combination of HPLC and mass spectrometry. I'll be showing you the image. It's in LCMS you get a liquid chromatogram, followed by, if the chromatogram has, let's say, 3 peaks, it indicates there are 3 uh, uh, 3 peptides being subjected, and these 3 peaks can have 3 mass spectra, because each peak will have its own respective mass spectra. It's like as it's moving to the column, every peak is indicator of a different size peptide. So this information, what we get from mass spectrometry can be used for studying the molecular weight or structural identity or quantifying the protein content in the given sample. So you can see the LCMS, how it works. Okay. So here, this is the point where your sample will be mixed with a mobile phase. Okay. So here, this is not a sampler unit where your sample is picked, and then you can use solvent system as acetonitrile or formic acid or methanol. It depends on what combination of solvents we use for separation of that particular protein set. So once the solvent system is standardized, this mixed solvent system will be mixing with a sample that is being injected or loaded through that auto sampler. Once it is mixed, then they are going to be passed through the column. This column. After they pass through the column, they will be passing through one by one peptides. As they pass through, each peptide in a LCMS, LC you get chromatogram, you may get LC chromatogram, and each chromatogram will be having its own mass spectra, will be having its own mass spectra, right? So once it passes through the mass spectra, you get, mass spectrometer, you get mass spectra. So normally when you do LCMS, you get chromatogram and mass spectra. So this is how the you can see here. You can see here there is chromatogram. The purple peaks are chromatogram, and each chromatogram have their own mass spectra. And this has its own mass spectra. So working of HPLC, once the HPLC sample is injected, it goes to the ionizing chamber from HPLC. From ionizing chamber, it goes to the mass analyzer. In mass analyzer, the peptides will either get fragmented and they get separated to the mass analyzer and then get detected at the detector. Then uh, uh, after gets detected, the mass spectra is generated. So there are mainly three components that you see in a mass spectrometry: ionizing chamber, then you have mass analyzer and detector. These are the three main components. These are the three main components that you see in a mass spectrometry. So ionization can be of two types. One is electrospray ionization. The other one is Maldi top or Maldi matrix assisted laser desorption ionization. So ESI, that is electrospray ionization technique, is the technique used to produce ions. It's important for the molecule that is being subjected to the mass spectrometry should be ionized. So you can either use ESI or you can use Maldi, which I have covered in my experience. So in case of electrospray ionization technique, the soft ionization technique developed by John Fenn in 1980. Here, the peptide sample will be passed through a very narrow orifice, very narrow hole, which will be highly charged. As it will be passing through, the the mixture will be passing through as a droplet, and this, because of the homogeneous charge that you see in the mixture, they get they will be fission. They will be fission. They get separated from each other, and then they enter into mass analyzer. Here you can see they are entering into the mass analyzer. So here the process of nebulization and charging takes place. Before they get into the mass analyzer, this cone will be heated up. This normally looks like a cone. This will be heated up. And as they pass through, they get desolvated, and then there will be evaporation. The ion enter into the mass. So once they enter the mass analyzer, I'll just talk about Maldi. So this is also kind of technique used for concentrating ion. That the proteins, I mean the peptides, should be converted into ionic form before they enter into the mass analyzer. So to convert them into ionic form, we use ESI or Maldi. I'm covering only two of them. So ESI and Maldi. So ESI, electrospray ionization, is how it does. Whereas Maldi, what we do is, this is the target plate of Maldi. You can get different types of target plate. Now you have to mix your peptide sample with the Maldi matrix. Okay? It can be positively charged matrix or negatively charged matrix. You can take it and mix it with the matrix. These matrix, once they are hit with a laser beam, this matrix gets dissolved. It becomes vaporized. And as it gets vaporized, the peptide sample also gets along with the peptide. So that is called desorption. Matrix assisted desorption. Desorption is reverse of absorption, or what you say, adsorption, surface phenomenon. The reverse of adsorption is desorption, right? So it's a matrix assisted desorption, uh, laser ionization, right? So once the laser is hitting the analyte along with the matrix, we place it on the Maldi target plane. So once the laser is hitting it, you see there is a kind of desorption process where there is matrix molecules are getting evaporated, and also along with that analyte also gets evaporated with it. So the moment it gets evaporated, there will be a strong positive charge that repels these charged evaporated molecules, through which they directly enter into the, once they are hit, and through strong cation electromagnet, these charged particles enter into the analyzer, that is ion. So I told you about how electrospray ionization, where the molecule under study gets charged. In Maldi, how the molecules get charged. So once the charged molecules enter into the analyzer, we should now understand what happens in the analyzer. Analyzer is a system where actually the charged ions move through the analyzer and get separated based on their mass and charge. Okay? Because these analyzers control the movement or the mobility of the ions through the analyzer tube. Now, controlling these charged ions, if they have higher charge, they move faster. If they have higher charge, higher mass, they move comparatively slower. The mass also plays a major role. So I would say the movement of these ions in the analyzer depends on their mass and charge. So we have different types of analyzer. One is time of flight analyzer. Another one is quadrupole analyzer. Quadrupole analyzer means four poles. Quadru, that quadra means four poles, where there will be change in the positivity and the negativity in the pole. So you can see here the change in the charge in the poles that's happening. And as the ions are moving through the poles, these change in the charge will control the movement of the charged ions or the charged peptides through these analyzers. Okay. Now, after they pass through the analyzer, they go and get detected. Now, depending upon the mass and charge, these get separated. Okay. Now, tandem mass spectrometry, also known as MSMS or MS square. Tandem means we have multiple analyzers. You can have top. You can have a top analyzer followed by another top analyzer. So two top analyzer, it is called top. You can have quadruple, you can have triple quad, where it's quadruple collision cell and quadruple, that can be a triple quad. So you can have multiple analyzers, calling them as mass, what do you call it, tandem mass spectrometry. Okay. Here you can see this is a triple quad, where you have quadruple followed by collision cell, again followed by a quadruple analyzer. Okay. You can have Q-top, which can be a quadruple time of flight combination. So you can have different types of combination analyzer depending upon what how efficient your work should be. Then comes the detector. Detector is the final part of the mass spectrometer, where it detects the charge of the ions that are hitting the detector. Okay. Now, if you see the detector, how does it work? Is the ions come and hit the dynode. The charge, this leads to the process of phosphorescence. through phosphorescence, the amount of light that is illuminated, that will be multiplied using a photomultiplier, and then the mass spectra are generated for that particular peak. So here is one more example of triple quad. This is the tandem mass spectrometer. You can see this is a LCMS. I told you normally LCMS is a combination of liquid chromatography. This will generate a chromatogram, and this is the mass spectrometry. This will generate the mass spectrum. So we have it in combination. This is a Maldi top. Okay. So this is where we load the target plate. This is the Maldi top analyzer. So as I told, how does it work? Creating of ions, separating of ions, detecting of creating of ions by ESIMS or Maldi, separating by using analyzers. It can be top analyzer, it can be quadruple analyzer, it can be Q-top, it can be triple quad, okay. And then detection of the ions. Final thing is database analysis. I told you that there are different types of mass analyzers. Q-top, sorry, time of flight analyzer, ion trap, then quadruple, Fourier transform, ion cyclotron, mass analyzer. Okay, once we get a mass spectra, how are we going to study and find out what this mass spectra is revealing? You just get a stick diagram in the mass spectra. Once you get the stick diagram in the mass spectra, how do you find out what it is actually giving the information about? You can see here, just taking this as an example. So this is a relative intensity of the peak. The higher the peak, the longer the peak, the abundance of that particular component or the peptide is high. Okay. So here we are doing about proteomics, and I discussed about gel separation and cutting the gel and digesting the gel. Once you get the trypsin digested fragments, you then load it onto the mass spectrometry. So each peptide will be fragmented into different peaks, but each peak has information to give you. So here, this we call it as, there is something called a molecular ion peak and then the base peak. The longer the peak, what you see here, it can be a base peak, and the peak that has the highest value and measurably high, that you can consider it as a molecular ion peak. Now, how do we find out that amino acid sequence that is present, the information of amino acid sequence that is present in the spectrum? Here, this is how we find out. If the M by Z value of alanine is 72, what might be the M by Z value of alanine plus valine? It should be 171. What should be the combination of M by Z value of alanine, valine, alanine? It should be 242. So it's a cumulative mass. So you need to look for whether you have the M by Z value of 171, whether peak is there at 171, at 72, and 242, and similarly 299, 402, 473, 530, 601, 769. If you are able to find out these peaks in the mass spectra, then this indicates that the sequence is as follows. So normally, depending upon the M by Z values or the peaks that you see in the mass spectra, we will be able to find out what is the peptide that is present. And looking at this peptide, we can go for bioinformatics and you can find out which protein this peptide is from. Once we come to know the protein name, we can further find out what role that protein is playing in the disease and then claim that it is a potential biomarker. So there are other techniques also, isotope labeling. I have worked on iTrack, which is called isobaric tag for relative and absolute quantification of proteins. It's also one of the techniques, but we don't use any gel in this technique. We directly take the sample and then we tag them with a specific tags, mass tags, 114 mass, 115 mass, 116, 117. We add them to the peptide samples directly, no gel, direct protein separation, cryptic digestion, addition of these mass tags to the respective sample, then cool them up. After you incubate them overnight, then you can cool it, pass them through a strong cation exchanger, okay, the reverse phase column, then you can do LCMS. Here you can actually resolve around 5,000 peptides in one step. High throughput technology, and then you can find out what are the differentially expressed proteins. Then if you find out there is a 10-fold difference or 7-fold difference of protein expression, then you can claim that protein to be a potential biomarker. Then comes the bioinformatics part where you find out the peptide identification. I told you how do we do that. Normally, now we have software. Software will identify the name of the peptide, and you need to study through bioinformatics what's the role of that particular protein. So I showed you how the sequencing is done. We have softwares to do this. You can use Proteome Discover. You can use a mascot, Sequest. They do database searching. They will find out the protein, and also they will find out what is the level of expression if you are doing iTrack. So you will be able to find out from which protein the peptide was from. You can locate the peptide fragment, which protein it was from. You can use mascot, freely available. There's one more technique called mass spectral imaging. Okay, this was technique which I was working in Europe. Mass spectral imaging. Now, currently in India, not many are using this technology, but this will give directly in tissue identification and expression level techniques. Thank you.
Thank you, sir, for a wonderful lecture. So,
Yes.
we have some queries from the participants. So, what will the effect of the dyes on the samples?
Which dye? Fluorescent dye.
They didn't mention any dye as such. Suppose if you are using dyes, so what will be the effect?
Nothing, nothing. They normally say it's about proteins, right? So proteins, these dyes are specifically going and binding to the proteins. They will not modify the sequence, or they will not have the sequence, or what you say. I don't know what is their concern. Effect in sense, what type of effect they are looking for. But they don't have, from the study point of view, in gel studies, dyes don't have any direct effect on the proteins.
Okay. Okay, sir. So, participants, others, if you have any queries, you can unmute yourself and you can ask. Sir, next question is, what is the range selected in the detector for analysis?
In the range?
What is the range
Yeah.
selected in the detector for analysis? Mass
No,
spectrometry.
it depends. Yeah, it can depend. It can depend from 200 to 1100. Sometimes it will be 600 to 200. Sometimes it will be 50 to 600. It depends, like what can be the maximum M by Z value that you can expect from your study. But what happens in proteomics, we cannot have a defined idea about which proteins are we studying on. So it's better to have 1,200 to let keep a wider range, because if you already know what you're studying, then you will know the defined range you can locate it. You can put the range of it. It's about the M by Z value that has to be covered. Right?
So next query is how a protein can be identified.
Protein, see, once you perform a mass spectrometry, you get a mass spectra, you get M by Z values. So we are not going to actually take the spectra image and work, but we take the M by Z values, which is a numeric form, right? So those values we are going to use and paste it in. You can use mascot, which is freely available. You can go to the mascot, the peptide fingerprint page. There you can paste your M by Z values. You can run. You will get the peptide, the peptide information that was there in the M by Z value. And that peptide you can do blast and find out to which protein it is matching. You can identify the name of the protein.
So the next query is can you explain the importance of docking in proteomics?
Okay. Well, let's talk about structural proteomics. If you are looking for structural proteomics, then docking has to do with the protein structures. Here we are talking about sequences, right? So in case of docking, you need to have structures. You first thing is you should identify the structure of the protein. To identify the structure of the proteins, you follow either three-dimensional NMR or cryo-electron microscopy, or you can use X-ray diffraction technique, or you can do theoretical modeling like homology modeling. Or once you do find out the structure of the protein, if you know them, then you can perform molecular docking. Molecular docking is a technique where you simulate the interaction between a drug and a protein. Right? That's it. So it's just a simulation. It's a theoretical approach where you try to hypothesize that this can be a mode of action.
Okay, sir. Next
Understand?
query is
So
can you
yeah.
tell us the best statistical methods to look at differences in protein profile from mass spectrometry result?
Can you repeat the question?
Can you tell us
Statistical.
the best statistical methods to look at differences in protein profile from mass spectrometry result?
Okay. Well, I had used the Sequest. Proteome Discoverer is a tool. Uh, what we do is the fold expression, it's like two-fold up regulation or two-fold down regulation. We check how much, what is the quantity, or once we quantify the peptide levels in a given sample. And when you compare it, you just compare whether it is high or low. The statistics is quite simple. Those we use online, I mean, commercial softwares. In case if you're talking about mass spectra result, in mass spectra we don't do use any statistics. Okay? But once you get the come to know the name of the protein and the peptide, then you can find out the abundance of the peptide. Then you can do the statistics there, comparative analysis. For that, you can use either Proteome Discoverer is appropriate, and Sequest and Mascot, these are the one which will find out the sequence of the amino acid.
Okay, sir. The next question is can we see exosome protein marker in MS spectra?
Exosome protein. Yeah, exosome. See, exosomes. First thing is you should be able to harvest exosomes. So whichever samples you are looking for, first collect the isolated exosome, and then do extraction of proteins. Then you can do sorting out. Then after sorting out, you can directly go for tryptic digestion and LCMS, or you can go for SDS page or 2D page. Then once you come to know that these are the spots that you are interested in, protein spots, cut them, excise them, perform mass spectrometry. Will come to know.
Okay. Okay, sir. Now I request our HOD, Dr. B.K. Manjunath, to speak few words.
Good morning, Joy.
Good morning, sir.
Thank you very much for your brainstorming talk on proteomics.
Thank you, sir.
Hope our students and participants enriched their knowledge with the proteomics. And I congratulate all the participants for participating in the webinar and updating their knowledge. And on behalf of Demand of Biotechnology, I wholeheartedly thank Dr. Joy for accepting our invitation and coming over here and presenting webinar on proteomics. And we look forward for the collaborative projects with Dr. Joy so that
Sure.
we can also enrich our knowledge with proteomics and genomics. Thank you, Joy, Dr. Joy, for accepting our
Thank you, sir.
invitation and coming
Thank
over
you so
here.
much.
Thank you.
So, on behalf of the management, staff and students of the Department of Biotechnology, the Oxford College of Engineering, I would like to extend my sincere gratitude to Dr. Jai for his excellent lecture today. Thank you so much, sir, for accepting our invitation and giving a wonderful lecture. Also, I would like to extend our heartfelt gratitude to our chairman, Sri SNVL Narasimha Raju, for his continuous support. And also I would like to extend our sincere gratitude to our principal Dr. Aravind A.S. and our director Dr. Amarnath K. for their support, continuous support. I would like to extend my heartfelt gratitude to our HOD Dr. B.K. Manjunath for his motivation, guidance and support in organizing this event. And also I would like to express our gratitude to Mr. Mukund, our alumni, for attending the webinar. And also I would like to extend our sincere gratitude to all the participants for attending the session, and also to my dear colleagues for helping in organizing this event, and a special mention to our student Mr. Rohit for his technical support. Once again I thank everyone for the kind participation in the webinar. Thank you, sir. Thank you so much. Sir, we will end the meet.